So the first experiment that we want to do is carbohydrate digestion, and we need 5 test tubes for that, labeled 1C to 5C. And the second material is the Romali ring solution, and we're going to be adding that to all of our tubes at the beginning of the experiment. And then the next thing is you need to have your pancreatic juice, which contains the alpha amylase and other enzymes. Then you also need the ionized water, and after that you need to have the alpha amylase, which is going to be used for positive control. You need to have the starch solution, which with the substrate in this case. Potassium iodide will be used to test for presence of starch in our sample after digestion. The Barfred reagent will be used to test for monosaccharides and disaccharides in the sample after digestion. And also we'll be using the Benedict solution to test for the presence of reducing sugars in our sample after digestion. So the first thing is to add 1 ml of mammalian ring solution to each of the test tubes. So you have 1000 microliter, which is equivalent to 1 ml, and you're going to be adding that to each of the test tubes. Immediately you're done with that, then the next thing is to take the deionized water, and you're going to add 1 ml of deionized water to tube 3 and tube 4. So after this, you're going to take that substrate, which is starch solution. Remember, starch is the carbohydrate that is stored in plants, and so you're going to have to add the starch, 1 ml of the starch to all of the tubes except tube 4. So we're going to add starch to all of the tubes, but not tube 4. Then the next thing we want to do is to add the source of our enzymes. Remember I said pancreatic juice contains the alpha amylase, and it's usually found in the duodenum, released from the pancreas, and then it's transported to the duodenum. So there we're going to add 1 ml of the pancreatic juice to tube 1 and tube 4. And then the next step is to add the alpha amylase to our to the tube 5, which will serve as a positive control. And remember, this is like the enzyme that's required to uh, digest starch and to form the end product, which is the maltose. So that's our positive control 5C. So take note of that, or 5A. And then after that, you mix your samples by vertexing them. So you have to vertex each one of them and make sure they are well mixed. And then after you mix them, you add paraffin on each one of them to prevent them from spilling over during incubation. So after covering the each of the samples tubes with, then we take um, tube 2 and you put it in the ice pack. So that's an ice water bath, and then you put it there while you take the rest of the samples into 30 degrees centigrade water bath, and we're going to allow it to stay there for the duration of the incubation, which is going to be for 1 hour. So we allow it to incubate for 1 hour, and then after that we're going to run our test.